Hi, I'm Minan Çortaç. DevaNano was formed to improve cancer therapy through our proprietary enzyme delivery method. Before I tell you how, I will tell you why. Can we cure cancer? Actually, yes, we know how to cure cancer. It may seem like a brave statement, but bear with me. Consider, 40 years ago, childhood leukemia, the death rate was 80%. After 40 years, now the cure rate is 80%. How this has been achieved? With a drug called L-asparaginase. Asparaginase is an enzyme that depletes the amino acid asparagine. Leukemia cells cannot live without it, but normal cells can't because they can produce their own. But why hasn't this been translated into other types of cancer? There are two main problems. One of the first important problems is mostly useful enzymes are from foreign origins, such as from bacteria or yeast. So they generate immune response. These immune responses causes a severe allergic reaction, and also these immune responses destroys the activity of the enzyme rapidly. The second problem is the loss of cofactors. I will tell you a story about this. 20 years ago, it was discovered that most 60% of tumors cannot salvage methionine. So, based on this, a local company called Anti-Cancer developed an enzyme called methionase. And it was a very safe enzyme, but however, it didn't work because of the cofactor loss. Because enzyme methionase required a cofactor called vitamin B6. And blood proteins was stealing vitamin B6 from methionase. So, our technology can solve both of these problems. Here's how. Our platform consists of a hollow nanoparticle where enzymes are encapsulated in the hollow interior, but they can't escape. However, at the same time, blood proteins or antibodies produced by immune system can't go through the shell and access the enzyme. Amino acids, however, can go through the shell and access the enzyme and can be depleted. So, how are we going to apply this? Imagine a patient with metastasized tumors. We have our enzyme-loaded shells, and we inject them intramuscularly, where they deplete amino acids systemically. Once depleted, tumor cells are starved to death, and they die selectively. We don't need to know where the tumor is, because tumor cells starve to death, but normal cells can survive because they can produce their own amino acids. Then the treatment is delivered to the tumors selectively. And this works actually. Here is in the left, you see where the particles are located at. And in the right, you see the luminescence generated by the encapsulated enzyme. And days later, activity still remains. And this is a picture of asparagine depletion with intramuscularly injected particles. However, if there is an existing immune response, there is no depletion at all. The entire activity is completely destroyed. If we encapsulate this enzyme into our nanoparticles, we not only improve the durability of the depletion, with the exact same immune response that destroys the activity of the free enzyme, doesn't do anything to the particles. This technology has developed in UCSD, and DevaNano has already licensed 3 key patents that cover this technology. And this technology depends on the unique synthesis method that we are working on. And this is a highly scalable, neutral pH, and liquid phase reaction that you can apply to any enzyme. The state of the art of the competition is a molecule called PEG. The enzymes are functionalized with PEG to get protected. However, this is an extensive and needs to be customized for every single enzyme, and it doesn't really prevent cofactor loss. Also, 25% of patients have an immune response against PEG. I'm not talking about the enzyme, against PEG. So this immune response can render the whole therapy useless. We are not the only nanotechnology trying to do enzyme therapeutics, though. But we are the only technology that can address all the requisite features to succeed in the clinic. And most importantly, we have the highest loading efficiency ever reported in the literature. Why is it important? Because we have 10 times better loading capacity by using 10 times less carrier material. This gives us 100-fold improvement over competing technologies. And we know, we are all familiar with silica. We walk on it at the beach, and it's a common food ingredient. So we will have three main initial products. First is aspergillus shells. With aspergillus shells, we will address pediatric and adult leukemia. With methionine-loaded shells, we can address 60% of tumors. With uric acid shells, we can address tumor lysis syndrome as well as chronic gout. But let me emphasize, in all these cases, the therapeutic mechanism of action is already established. 95% of drugs fail in clinical trials. But by using already approved, validated therapies, we take away the majority of that risk. To achieve this, we are asking $2 million for the first round to show the preclinical proof of concept of these three indications. Later, we will ask $10 million to carry us towards the clinical trials and IND. And we will do another Series C round for human clinical trials. Also, we are offering multiple exits to the investors, which I can go into detail after the presentation. This will be achieved by a team that is led by Dr. Abe Gaila, who is a serial entrepreneur and has started multiple companies with successful exits. And I am the inventors of shells technology, and I will oversee their development. And our cancer biology and preclinical testing will be taken care of by Dr. Bradley Mesmer. This team is backed up by a world-renowned scientific advisory board, including Nobel laureate Dr. Roger Chen, who is an expert in translational biomedical technologies. Dr. Dennis Carson is the former director of Moores Cancer Center, and he's an expert in clinical trials and cancer therapeutics. Dr. Sadik Ezner is an expert, world-renowned expert in nanotechnology, and he has started multiple successful ventures previously. DevaNano has a unique platform for enzymatic therapies, and our technology represents the next generation in enzyme encapsulation, which will drive the development of best-in-class and first-in-class pharmaceuticals. With so many verticals, we will give a lot of opportunities to early investors. Strong IP, strong scientific team, backed up with a world-renowned advisory board, complemented by smart money investors, will help DevaNano help patients with cancer. Thanks for your attention.
Okay, do we have any questions or comments from our judges?
So you'd mentioned the Series A money of $2 million,
yes,
which would culminate with a proof of concept study. What's the nature? What is the killer experiment or the proof of concept study that would come out of that?
Actually, maybe I have a slide that breaks it up. So this is basically what it is. So, for instance, right now there are three directions that I'm talking about. Aspergillus therapy is a well-established therapy, but there is a problem with immune responses. One of them is to make sure that we will not create that immune response that is seen in the clinic. For methionine case, there are many directions that we can apply the cancer. And this includes colon cancer, prostate cancer, lung cancer, many directions. So we will validate the technology with different types of cancer, which will also be one of the major efforts. Also, uricase for chronic gout is also well-validated technology, but we need to also make sure that the immune responses are prevented, as we are seeing in animal studies right now. We need to further improve it.
How has the company been funded to date? How much went in? And kind of what is the business process of what you're doing? We heard about the scientific process, but what is the business?
So business process, actually, so right now, we have some seed funding, and with this proof of concept studies, then we will have multiple directions. We can use some of the initial results to license our technology for certain indications and get some revenue to the company that can fund other technologies or can also reward the early investors as well. So right now, in a few years, we are expecting that there can be some return to the investors. I mean, so far we have around $100,000, which covers some of the early results that I have mentioned, and also we have some grant money that basically this is a part of my PhD project, and this was we have some grants from NIH, and so far it was funded with those grants, and we have already licensed our key patents. So right now we are looking into moving forward. Since we have the proof of concept studies, now we are moving forward towards working with actual xenografts and tumor models.
So a couple of questions. The first one is, a lot of times people think that focus is really important. I mean, investors come to companies and say you need to focus. The fact that from the beginning you're going to start 3 programs is a little concerning to me, up front, just looking at the whole picture. What are your thoughts on that?
That's a very good question. But here, the key is actually this is one of the good parts of this technology. Basically, whatever we do for all these 3 different applications, they're all based on the same technology. So whatever we develop for them, that can be applied to other technologies as well. And that was one of the reasons why we chose the 3 indications for our first try. Because we already know the enzymes work, but they have some complications that I'm talking about, such as immune response or cofactor loss. And we know that with the same exact chemistry, we can solve those issues. So our basically business plan is to apply validated technology that in that sense, in terms of technology improvement, they're all connected. But it will involve more experiments to be done. That justifies our initial ask for $2 million. Exactly.
Yep.
So then,
asparaginase is a drug.
Yes.
So it will be pretty straightforward to you to understand what the market for asparaginase is, how you can replace that market, and the risk it will be lower initially.
Yes.
Okay, as long as you can deliver and there's no homogeneity, it will work.
Yeah,
so
exactly.
I think that that would make the pitch more interesting, more attractive for me because it will be more focused.
Yeah, but actually that's a very good point. So this is how we lined up our indications actually. So asparaginase is very loaded, it's already established, and we already have very nice data with that, so then we can build on it. But it is relatively smaller market than the others. But methionine is a huge market, and it was a promising technology, but it failed due to cofactor loss. So this is like a leader. Also uricase. Uricase is also validated technology. So these 2 technologies are our initial goals, but the data that we gather from them will also help to improve the methionase therapy, which can target a really big market. So in that sense, there is like a chronological order in that. So as you pointed out.
I'm not to get too technical, but a lot of people are doing nanotechnology and nanoparticles these days.
Yeah.
How is this differentiated without getting too technical for me or for the audience?
So basically, the beauty of this technology is, So, for instance, one direction is we do intramuscular injection. Intramuscular injection, then you don't need to worry about circulation or distribution of the particles. You know where they are and you know that. But you need to achieve a systemic effect by using those particles. To achieve that systemic effect, you need to have very high loading efficiency. And you need to go too much in toxic because there are many different materials and they have different toxicities. And if you're talking about enzyme, you need to have very high enzyme loading with very little material because in the end you will reach the toxicity limit. But our technology is the only technology that allows that much loading efficiency, which can even work in a localized setting. Otherwise, they inject the enzymes and they circulate. But with our particles, we enable them to stay in a manageable, so it offers a manageable distribution and circulation kinetics, but at the same time, it can achieve the depletion. So it can achieve the effect. In that sense, we don't see too much risk going into the clinic. So I think this is one of...
You suggested that there is no immune response. Would you articulate a little bit more this matter? Because with RGNS, the frequent problem is immediate hypersensitivity response. And so I do not understand, since the enzyme leaks out from the nanoparticle, how do you avoid any immune response that might occur? Could you perhaps address this matter?
Actually, that's a very nice question, but probably I couldn't have time to address the technology part of it. The beauty of the technology is we don't release the enzyme. So enzyme is always inside a cage. It never leaves. So antibodies or other blood process cannot access it. Throughout the process, enzyme stays hidden inside the particle. The only thing that goes through is the amino acids. Because it's like a cage, the amino acids can go through the cage and access the enzyme. So since it is not exposed, then it doesn't really generate an immune response as long as it's encapsulated. And eventually, since the particles degrade, it may leave, but you already cured the cancer, so hopefully. So.
If the enzyme that presumably is consumed by T cells, let's say in the case of T cell leukemia, the T cells use the arginine, and presumably you try to deplete that arginine with the
Arginine, yeah.
So the enzyme would get out in order to accept its effect, otherwise it would be ineffective.
No. Enzyme substrate is smaller, so it's like a nanoporous cage. So once sealed, maybe I can go through the technology. So
Actually, I'm going to have to just stop right there.
Oh,
You have to
okay.
stay on schedule.
Okay, yeah.
Okay, let's hear
Sure.
it one more time for
Hopefully,
Maybe I
DevaNano.
can-